Hello friends, warm welcome in this video, that is innate immunity, in which we will learn physiological barriers. Friends, in the earlier videos we have seen the various components of innate immunity. The various innate immunity barriers includes anatomic or physical barriers, which includes skin or mucosal membrane. Then physiological barriers, which we will study in this video. Then phagocytic or endocytic barriers. We have already learned in the earlier video what is phagocytosis and what are the different cells involved in the phagocytosis, and the inflammatory barriers. We have learned the various cardinal signs of the inflammatory response. So in this video we will learn what are the physiological barriers. So friends, physiological barriers may include temperature, pH or chemical mediators. The chemical mediators involves lysozyme, interferons, complement factors, toll-like receptors and various other chemical mediators. So we will learn one by one. Friends, certain species of animals are more susceptible to a particular disease, whereas there are some species which are at all protected from a particular disease. For example, in case of birds or in case of chicken, the body temperature protects them from some infectious diseases like anthracis. So chicken is protected from the anthracis. The gastric pH is also one of the important physiological component which protects some species from particular infections. The low pH protects adults from certain diseases like pneumonia, meningitis, conjunctivitis, candidiasis and many more. The gastric pH is one of the factors which protects adults from certain diseases and certain diseases which are common in the newborns. So the chemical mediators includes lysozyme. Lysozyme is one of the enzyme which is responsible for cleavage of beta-1,4 linkage between N-acetylglycosamine and N-acetylmuramic acid of peptidoglycan linkage. Peptidoglycan is the component of bacterial cell wall. So lysozyme kill bacteria by cleaving this peptidoglycan. Friends, in the earlier videos we have learned defensins and cathelicidins are being important barriers such as on the skin and the epithelial layers connected to the body's opening. Friends, some cells and tissues constitutively express these peptides. For example, human intestinal panepithelial cells constitutively express alpha-defensin and some beta-defensin. In addition to this, some beta-defensins and cathelicidin LL37 are constitutively synthesized and packaged in the granules of neutrophils, which are ready to kill phagocytosized pathogens like bacteria, fungi, viruses and protozoans. In some cells like mucosal and glandular epithelial cells, skin keratinocytes and natural killer cells, the expression of this antimicrobial peptide is induced or enhanced by signaling through pattern recognition receptors, particularly toll-like receptors. Macrophages do not produce these antimicrobial peptides. However, there is an indirect pathway by which microbes induce cathelicidin in macrophages. Binding of microbial ligands to macrophage toll-like receptors induces increased expression of receptors for vitamin D. Binding of vitamin D to these receptors activates the macrophages to produce cathelicidin, which then can help the macrophage kill the pathogens. Another major class of antimicrobial proteins transcriptionally induced directly by pattern recognition receptors is the type 1 interferons, which include interferon alpha and beta. Type 1 interferon production is generally activated by those cell surface toll-like receptors and intracellular toll-like receptors that recognize viral nucleic acids and other components and activate the IRF transcription factors. Type 1 interferons are produced in the two situations. When virus infected, many cell types are induced to make interferon alpha and or interferon beta following binding of cytosolic viral pathogen associated membrane proteins such as nucleic acids to their receptors. In addition, many cells express cell surface toll-like receptors recognizing viral PAMs and or internalize virus without necessarily being infected, allowing endosomal toll-like receptors to recognize viral components. Signaling from these toll-like receptors activate the IRFS and interferon alpha and beta production. One particular type of dendritic cell, which is called as a plasmacytoid dendritic cell, because of its shape, is particularly effective producer of type 1 interferons. The plasmacytoid dendritic cell endocytosis virus that has bound to various cell surface proteins. Toll-like receptors 7 and 9 in the endosomes then are activated by viral PAMs leading to IRF activation. Type 1 interferons exert their antiviral and other effects by binding to a specific receptor, which are frequently called as IFNAR, which means interferon alpha receptors, expressed by most cell types. Similar to signaling pathways downstream, many cytokine receptors binding of interferon alpha or beta activates interferon alpha receptors to recruit and activate specific JAKs, which then activate specific STATs. STAT1 and STAT2 dimers then induce expression of various genes, including those for the 3 proteins that block viral replication, which includes protein kinase R or PKR, which inhibits viral and cellular protein synthesis by binding the elongation factor E initiation factor 2 alpha 2 prime 5 prime oligoadenylate A synthetase, which activates the ribonuclease RNA gel that degrade viral mRNA, and MX proteins, which inhibits both the transcription of viral genes into mRNA and the assembly of virus particles. Reflecting the potent antiviral activities of type 1 interferons, they are used to treat some viral infections such as hepatitis B and C. In addition to their key roles in controlling viral infections, type 1 interferons have other immune-related activities in that they activate natural killer cells and regulate activities of macrophage and T cells. Treatment with the interferon beta has been shown to have beneficial effects in some forms of multiple sclerosis, a T cell mediated autoimmune disease with inflammatory involvement, probably by inhibiting production of certain pro-inflammatory T cell derived cytokines. Cytokines are the proteins transcriptionally induced by pattern recognition receptors activations, which, while not directly antimicrobial, activate and regulate a wide variety of cells and tissues involved in innate, inflammatory and adaptive responses. Cytokines functions as the protein hormones of the immune system produced in response to stimuli and acting on a variety of cellular targets. Here in this table, various cytokines induced by pattern recognition receptors activation during innate immune response are listed along with their effects on the target cells and the tissues. 3 of the most important cytokines are interleukin 1, interleukin 6 and tumor necrosis factor alpha, which are major pro-inflammatory cytokines that act locally on blood vessels and other cells to increase vascular permeability and help recruit and activate cells at site of infection. They also have systemic effects. Interleukin 1, interleukin 6 and GM-CSF also feedback on bone marrow hematopoiesis to enhance production of neutrophils and other myeloid cells that will contribute to pathogen clearance. Activation of monocytes, macrophage and dendritic cells through some toll-like receptors also induces production of interleukin 12 and interleukin 18. The cytokines that play key roles in inducing naive helper T cells to become Th1 cells, in particular by inducing production of interferon gamma. The hallmarks cytokine of Th1 cells is interferon gamma, which stimulates cell-mediated immunity and is an important macrophage activating cytokine. Hence, interleukin 12 and interleukin 18 are also considered pro-inflammatory. Interleukin 10 is another important cytokine specifically induced by some toll-like receptors in macrophages, dendritic cells, other myeloid cells, and subsets of T, B, and natural killer cells. Interleukin 10 is an anti-inflammatory in that it inhibits macrophages activation and production of pro-inflammatory cytokines by other myeloid cells. Interleukin 10 levels increase over time and contribute to controlling the extent of inflammation caused tissue damage. Chemokines are small proteins which act as chemoattractants, means agents that induce cell to move towards higher concentration of the agent, recruit cells into within and out tissues. Some chemokines are responsible for constitutive migration of white blood cells throughout the body. Other chemokines produced in response to pattern recognition receptors activation have key roles in the early stages of immune and inflammatory responses, in that they attract cells that contribute both clearing the infection or damage and to amplifying the response. The first chemokines to be cloned, interleukin 8, which is also called as CXCL8, is produced in response to activation by PAMs, DAMs, or some cytokines of a variety of cells at the sites of infection or tissue damage, including macrophages, dendritic cells, and epithelial cells, as well as vascular endothelial cells. One of interleukin 8's key role occurs in the initial stages of infection or tissue damage. It serves as a chemoattractant for neutrophils, recruiting them to sites of infection. Other chemokines are specifically induced by pattern recognition receptors activation of epithelial cells in certain mucosal tissues and serve to recruit cells specifically to those sites where they generate immune response appropriate for clearing the invading pathogen. For example, B cells are recruited to the lamina porphyria, the lymphocyte-rich tissue under the intestinal epithelium, by two chemokines, CCL28 and CCL20, produced by intestinal epithelial cells activated by PAMs binding to their toll-like receptors. These activated epithelial cells, as well as local dendritic cells activated by PAMs, also produce cytokines that stimulates the B cells to produce immunoglobulin A, the class of antibodies most effective in protecting against mucosal infections. The complements are a set of serum proteins that cooperates with both the innate and adaptive immune system to eliminate blood and tissue pathogens. Like the components of the blood clotting system, complement proteins interact with one another in catalytic cascades. Various complement components bind and opsonize bacteria, rendering them susceptible to receptor-mediated phagocytosis by a macrophage which express membrane receptors for complement proteins. Other complement proteins elicit inflammatory response, interface with components of the adaptive immune system, clear immune complexes from the serum, and are eliminate apoptotic cells. A membrane attack complex or MAC assembled from complement proteins directly kills some pathogens by creating pores in microbial membranes. The biological importance of complement is emphasized both by the pathological consequences of mutations in the genes encoding complement proteins as well as by the broad range of strategies that have evolved in the microorganisms to evade it. The complement kills the pathogen by three major pathways, which includes classical pathway, lectin pathway, and alternative pathway. The last but not least component of a physiological barrier is toll-like receptors. Toll-like receptors were the first family of pattern recognition receptors to be discovered and are still the best characterized in terms of their structures, how they bind PAMs and activate cells, and the extensive and varied set of innate immune responses that they induce. Characterization of toll protein revealed that its cytoplasmic signaling domain was homologous to that of the vertebrate receptor for the cytokine interleukin 1. Intensive work over last decade and half has identified multiple toll-like receptor family members in mice and humans. As on 2011, 13 toll-like receptors that functions as a pattern recognition receptors have been identified in mice and human. Toll-like receptors 1 to 10 are conserved between mice and humans, although toll-like receptors 10 is not functional in mice, while toll-like receptors 11 to 13 are expressed in mice but not in humans. Each toll-like receptor has a distinct specificities for conserved PAMs. The toll-like receptors and some of their known biochemical studies have revealed that the structure of toll-like receptors and how they bind their specific ligands. Toll-like receptors are membrane-spanning proteins that share a common structural element in their extracellular region called as leucine-rich repeats or LRRs. Multiple leucine-rich repeats make up the horseshoe-shaped extracellular ligand binding domain of the toll-like receptors polypeptide chain. When toll-like receptors bind their pathogen-associated membrane proteins or damage-associated membrane protein ligands via their extracellular LRR domains, they are induced to dimerize. In most cases, each toll-like receptor dimerizes with itself, forming a homodimer, but toll-like receptor 2 forms heterodimers by pairing with either toll-like receptor 1 or toll-like receptor 6. Toll-like receptors exist both on the plasma membrane and in the membranes of endosomes and lysosomes. Their cellular location is tailored to enable them to respond optimally to the particular microbial ligands they recognize. Toll-like receptors that recognize pathogen-associated membrane proteins on the outer surface of extracellular microbes are found on the plasma membrane, where they can bind these PAMs and induce responses. In contrast, toll-like receptors that recognize internal microbial components that have to be exposed by dismantling or degradation of endocytosized pathogens, nucleic acids in particular are found in endosomes and lysosomes. Unique among the toll-like receptors, TLR4 has been shown to move from the plasma membrane to endosomes or lysosomes after binding to lipopolysaccharides or other PAMs. In addition to microbial ligands, toll-like receptors also recognize damage-associated membrane proteins, endogenous components released by dead or dying cells or damaged tissues. Among the DAMs recognized by plasma membranes, toll-like receptors are heat shock and chromatin proteins, fragments of extracellular matrix components such as fibronectin and hyaluronin, and oxidized low-density lipoprotein and amyloid B. While self-nucleic acids usually do not activate the intracellular pattern recognition receptors under certain circumstances, such as when bound by anti-DNA or anti-chromatin antibodies in individuals with autoimmune diseases, lupus erythematosus, they can be endocytosized and activate endosomal or lysosomal toll-like receptors, contributing to the disease. Thank you.